As I was saying earlier, we are going to take a look at the mechanism of action of two enzymes in addition to what we were doing the last time. And then we continue with enzyme kinetics. Then we continue to see application of enzyme kinetics in enzyme inhibition and a few applications of enzyme inhibition. So then we'll end today's lesson on that. Then the next time when we meet, we will look at, I think we had the last lecture we'll have in enzymes is going to be on the structure and function of some common coenzymes. So that will close the session on enzymes. After that, then we move on to the metabolism of biomolecules, that is carbohydrates, proteins, lipids, and nucleic acids. Of course, when we are doing metabolism of these biomolecules, you are going to build on the knowledge you have acquired in enzymology, because then a lot of enzymes will be acting in those biochemical reactions. So from time to time, your attention will be drawn to the types of reactions which are occurring there and how you can easily identify the types of enzymes that are participating in those reactions. Right, so what you see on the screen is a step-by-step mechanism of action of the enzyme chymotrypsin. Now, right from the beginning, let me caution that you don't need to cramp this thing into your head. So this is to let you appreciate the fact that even though enzymatic reactions are occurring at a really fast rate, and we talk of them as if it is a single event, a reaction, an enzyme occurring. Actually, these reactions occur in a step-by-step manner. So sort of a cascade of reactions occur in order to achieve a specific product to be produced out of such enzymatic reactions. So it's not for you to be cramping into your head, like the moment you see it. I know that sometimes you can be intimidated by how the whole screen is looking with a lot of things, but we'll take it one after the other and just for you to appreciate step-by-step activity of enzymes. And you want to also appreciate the fact that the diagram we are looking at, we are focusing on what is happening in the active center of the enzyme. We referred to the active center earlier on. That is the specific portion of the enzyme that is having interaction with the substrate. So that is what we are looking at. Now, when we were looking at classification of enzymes, specifically when we were talking about enzyme specificity, we mentioned chymotrypsin as one of those examples with group specificity. And the type of bonds that chymotrypsin breaks is a peptide bond that is formed between the carboxylic group of an aromatic amino acid and any other amino acid. So which means chymotrypsin specifically recognizes that bond that is formed with aromatic aromatic amino acids from the carboxylic acid side, forming the bond with the amine side of another amino acid. That is what we looked at. So you can go back and refer to that after this lecture. You look at that when we talk about the specificity. Either it is bond specificity or group specificity. You will see that example over there. So now we are looking at chymotrypsin itself. So the blue structure you see, the round blue structures that you see, that is the whole structure of the enzyme. So this is just some pictorial presentation of this, just to explain what can happen in this thing. So it's just a generated computational image just for explanation of what happens in the active center of the enzyme. Now, the white portion, unshaded portion of the enzyme is the active center of the enzyme, and sort of a pocket that is inside the molecule. And you see that this portion is being labeled like there are different portions of it. So we have, of course, the whole portion, the whole white portion inside is the active center, and that active center has some specific parts. So these small parts, the small groove here, is being labeled oxyanion hole, and then you have one large portion here, which is being referred to as hydrophobic pocket. Then what you also see is that there are some amino acids which are shown here. So you remember that I said the active center is usually made up of some 2 or 3 amino acids that are close to each other in such a way that they interact with the substrate, to have the reaction of that substrate, being catalyzed, so that the reaction will be sped. The rate of it will increase. You saw when we were looking at the energy profile that first of all, the enzyme binds to the substrate to form an enzyme substrate complex. And then that enzyme substrate complex, you have the activation energy to the transition state reduced so that the reaction can occur at a faster rate. So this is what is happening here. You have these amino acids that are in the active center. One of the amino acids you see here is serine, is being depicted SER, and there's a number on top of it, 195. That one too, I explained to you that when you see things like this, it only means that if you take a protein molecule, and that protein molecule is having, some number of amino acids in it, so that if you are counting the amino acids that are bonded one to each other, on the 195th position, the amino acid there is serine. That's what it means. You also see histidine also represented here. So the whole molecule is not being shown. So it's just the side chains that are being shown. Serine has a side chain of CH2OH. That is the reason why you see that the side chain is just being shown with this OH here. And intentionally, this red, the hydrogen of the hydroxyl group on the side chain of serine is in red, just to actually show the track of that hydrogen on the side chain in that hydroxyl group of serine. So we'll see along where the hydrogen is going. The histidine, the side chain, yes, is having an imidazole ring. And then there is also aspartic acid. The histidine is at the 57th position. You see the number there is 57. And you also see the aspartic acid is at, I think, 302nd position. So this is just to show the positions where these amino acids are, but because the protein chymotrypsin, that polypeptide, is folded such that these amino acids are close to each other, that they begin to have interaction between themselves, actually acting as the active center of the enzyme. Now, having seen that, okay, there is also some extra amino acids that may also be that are also having some interaction with the substrates. And here we have glycine at 193rd position, or 193rd position. And you have serine, another, so it's the same serine here, 195, 195. So they just showing it from another angle down there. Okay, now what also you see is that we have the reactions being numbered. So you have reaction number 1. So the reaction is actually occurring clockwise. The series of reactions that are being shown is numbered clockwise. So this is 1. So which means that this is the free enzyme, and that free enzyme will bind to this substrate, the protein or the polypeptide, which is shown on top of the first arrow, number 1, where you see that there's a portion of that protein which is in blue fonts, and the other side is black. Now, the black side is what is having the phenylalanine. So you see the aromatic ring, and on that carboxylic acid side of that phenylalanine, it is forming a peptide bond with the blue side of the peptide. So that's another amino acid in blue. And then you see that AA there, that shows that there are a number of amino acids still, on the left side. And of course, the AA on the right side too means that there are more amino acids also on the right-hand side. But the bond which is to be broken is between the black portion and the blue portion. That is the amide linkage, or the peptide bond, that is to be broken. So from reaction 1, then reaction 2 will occur, then 3, and then 4 goes to 5, 6, 7. That is when the substrate dissociates away from the enzyme, and then the free enzyme, can go into another cycle of a reaction. Now, what do we see? You see that this substrate, the polypeptide, which is having the phenylalanine in it with the aromatic side chain, when it binds to the enzyme, so now we are looking at the first reaction. When it binds to the enzyme, so now you look at the second image. When it binds to the enzyme, you now have the enzyme substrate complex being written ES complex. Now, what you notice is that the aromatic ring falls into that hole. That is the hydrophobic pocket. So the empty enzyme is having that hydrophobic pocket, and specifically into that hydrophobic pocket, you have the aromatic ring falling directly into it. So you can see that the molecule actually is, slightly stable. This complex is slightly, stable, so that it can now move to the transition state. Now, the mechanisms of reactions which we looked at earlier, one of them was acid-base catalysis. You have these reactions inside the active center of enzymes, depicting acid-base interactions, such that you have what we refer to as proton transfer. In other words, the transfer of a hydrogen ion from one amino acid onto the other in the active center. And that sort of proton transfer results into the activation of the enzyme, so that the enzyme now, proceeds to catalyze the specific reaction that is to be to be catalyzed. So you see what is happening here, that you have, say, that serine residue, which is having a hydroxyl group on it. The hydrogen of that hydroxyl group is having some call it some hydrogen bond or electrostatic interaction with the histidine, also in the active center. So that is represented with some dashed blue lines. And you see that there is some lone pair of electrons also shown with red dots. And these are having some sort of a nucleophilic attack on the carbon of the carboxylic acid group of phenylalanine. you see that looking at serine also from another angle, there you have the amine group. So that is the end. So here you have the hydroxyl group, which is the side chain. And down here, it is showing the amine group, the hydrogen of the amine group of the same serine having interaction of the oxygen of that carboxylic acid group. Now, you see, what you notice is that you have this molecule that you have these amino acids that are inside an enzyme having interaction with some specific portions of it. The next stage that you see is that that hydrogen of the hydroxyl group of serine is transferred from it. So we have now moved to the second reaction. So we are looking at the short-lived intermediate, the first short-lived intermediate written there as acylation. You see that the hydrogen is transferred from the serine onto the nitrogen of the histidine, that imidazole ring. And that hydrogen, the red hydrogen, is now having an interaction with the nitrogen of the blue side of the molecule. Now, you see also at the same time, serine is still having interaction with the oxygen of the phenylalanine. At the same time, one other amino acid, glycine, which is at the 193rd position, is also having interaction with that same oxygen. Now, think of it this way. It's like these two molecules, like you have the enzyme as a protein, and then the protein which is supposed to be broken. Now the enzyme holds two parts, like the two ends of the bond that is supposed to be broken. So if you see the bond which is supposed to be broken, that is between the blue side and the black side. You see that one side, which is the carbon, is held by the enzyme. Some amino acids hold that end. And then you have histidine also holding the amine side. So just like how if you want to pull something apart, you want to break something, you hold the ends and pull them apart. That is logically, that's what is happening. So you see that, yes, now the bond which is supposed to be broken, the two sides are being held. And once you have such interaction between these two ends, which is strong enough, pulling this bond apart, that bond gets broken. And now you see that the third reaction you see is being written there, first product. And that first product is the other, amino acid with the other, side amino acids left at the end, the blue side. So the blue side is broken off. And see what? As the blue side is broken off, it is gone with that hydrogen which was red, which was donated from the serine. So it goes with that first product, which is the blue side of the amino acid. You are left with just the black side, which is having the phenylalanine in it. Now, which means the serine residue has lost one hydrogen, and that hydrogen is from its side chain, the hydroxyl group. How does it gain that hydrogen back? So this is where water comes in. So in this reaction, water is playing a part where water comes in, and intentionally one of the hydrogens is also written in red so that you can see clearly how this hydrogen goes back to go and be accepted by the side chain of the serine. So as water comes into the reaction, you see the hydrogen first is having an interaction with that nitrogen of histidine. Remember, it is that nitrogen of histidine that donated the histidine donated that hydrogen from serine to the amine group, and it was broken off. So when water comes in, water comes to interact with that histidine, that nitrogen on the imidazole ring of histidine. Then you see that the hydrogen is now giving off onto the nitrogen, and histidine, gives back that hydrogen to the serine residue. You see the hydroxyl group. So take it that the water breaks into hydrogen ion and hydroxyl ion. So the hydroxyl ion binds to the part of the protein which is left in the enzyme, so that it becomes a full molecule, to complete the carboxylic acid side of that residue which is left in the active center of the enzyme. And serine also gains back its hydrogen. The next thing when you go forward is that after the hydrogen goes back onto serine, and serine is now whole again, which means that the enzyme is ready to get dissociated from the product. So the second product comes out, and you have that second product with the ring phenylalanine on it. The enzyme is free again. So the enzyme can go into another cycle of reaction. Now, as I said from the beginning, all what I'm saying, I don't expect you to be cramping all of these things into your head. I've told you from the beginning, this is just for you to appreciate the fact that enzymatic reactions occur stage by stage. Even though it's a very fast reaction, the reaction does not just occur, as a single event. You may be wondering, how can you be studying some of these things to really know the step-by-step way of how these enzymes act? Actually, now it's even easier to do that. But even before then, I can say that this is where the use of inhibitors come in. So if you use inhibitors to slow down a reaction, so that at different points, at different times, maybe if you are doing it within a period of every 1 or 2 minutes, you take a sample out of the reaction, and then you run it in maybe some nuclear magnetic resonance, or maybe plus some infrared. You'll be able to identify the structures that are present at different times in the reaction mix. But now even is much easier, because you can use computational modeling to actually study the sort of energy interactions between molecules. If you can do this modeling, usually we use methods like molecular mechanics and molecular dynamics to study some of these things. In fact, if in future, some of you may be interested in doing some computational chemistry, which is great, because you'll be able to model molecules and see the sort of interactions between them. Look at the energy levels at different stages of reactions. You can do that with there are a lot of softwares out there which with some training you'll be able to make use of them to calculate energy levels of different intermediates, and begin to predict that this is the kind of reaction, or the path that a reaction can take. So like I said, it's for you to appreciate that step by step. So please don't go cramping this thing into your heads, but at least you should see it, and you should have a feel of it, and know that these things are possible. If you have the slide, you can. I mean, this should not take you so long for you to actually look at what is happening. Just try to see if you can understand anything in this, reading the small notes on it. Of course, I'm not going to be asking you that describe how chymotrypsin acts on proteins to break bond between to break peptide bonds in polypeptides and that sort of thing. No, don't expect that from me. I will not do that. At your level, there's no need for that, but at least you should be exposed to it and get to know these things. All right, so this is a mechanism of action of chymotrypsin. We'll move and look quickly at mechanism of action of lysozyme. So lysozyme was also one of those enzymes that we saw as one of the examples when we were studying the classification of enzymes, specifically depending on the name, the reaction that the enzymes catalyze. So from the name lysozyme, you can tell that this enzyme is involved in the breakdown of some bonds, but specifically lysozyme breaks the glycosidic bond that is formed between the monosaccharide derivatives that form the cell walls of bacteria. So basically the cell walls of bacteria are made up of the polysaccharide peptidoglycan. And peptidoglycan has the monomers N-acetylglucosamine and N-acetylmuramic acid. So it's being depicted here that so this is a peptidoglycan molecule that is drawn at the right hand side, and it's even zoomed for you to see the glycosidic bond that is between N-acetylmuramic acid that is on the left hand side, and then you have N-acetylglucosamine with a glycosidic bond between these two monosaccharide derivatives. You have some participation of water in that reaction because it's a hydrolytic reaction. So here too you see water, participating in the reaction in breaking the glycosidic bond between these two monosaccharide derivatives, which are in peptidoglycan, the molecule that forms the cell walls of bacteria. So I mean, this one too is you can see it in stages that well, there are two different models. In the first model here too, you pay attention to the amino acids that are depicted in the active center of the molecule. So in the active center, you see glutamate, which is at the 35th position, and you also see aspartic acid, which is at the 52nd position. Now, the only difference between these two, some slight differences, is that here you see there is some nucleophilic attack of this oxygen carrying the lone pair of electrons on that bond is forming with the carbon 1 on muramic acid. You see, but you see that the hydrogen that is in the carboxylic acid group of glutamic acid is also, having that sort of electrostatic interaction with the glycosidic bond. In both cases, you see that happening. This hydrogen, which is on the carboxylic acid group of glutamic acid in the active center, you see that it is having interaction with a glycosidic bond. In the second model, you see that aspartic acid at the same time, the carboxylic acid group is also, having that nucleophilic attack on the carbon 1 of muramic acid. So the difference you see is that here, the second part, you see aspartic acid that is having that nucleophilic attack on the first carbon 1 of muramic acid that is forming a glycosidic bond. But on the left hand side, you see that's not happening, but rather you see that the lone pair of electrons that is on that oxygen there is having that nucleophilic attack on the bond that it is forming, call it, transfer of some charges onto the carbon, which, causes some sort of a resonance effect, which later in the second, if you see the product which is being formed is some carbanion. So you see that the bond there becomes some sort of, let's say it's carrying some charge. After you see the first product, which is the N-acetyl glucosamine broken off. And you see clearly that the hydrogen from the glutamic acid in the active center, goes with that first product. You see the same thing, the second part, that hydrogen from glutamic acid in the active center goes with the second product, which is N-acetyl glucosamine. And then you see, still aspartic acid in the product on the right-hand side is having some covalent intermediates, is forming a covalent bond with that first carbon. You go ahead and you see that, again, water comes in. So water comes in, water breaks into hydroxyl group, hydrogen group, the hydroxyl, and then the hydrogen ion. And so you see from here, the glutamate or the glutamic acid, takes back its hydrogen from the water molecule. And then the next thing you see is that the hydroxyl group from the water is now added onto the second product, which is N-acetyl muramic acid. So again, it's a step-by-step, process by which the bond is broken, the glycosidic bond between these two derivatives of monosaccharide is broken. So, I mean, what's the significance of this? Lysozyme, if it's breaking bacterial walls, if it's breaking these bonds that are between the derivative monosaccharides that are forming the bacterial walls, it means that it can serve as a bactericide. So it can, because once the cell walls of the bacteria is broken, it results into cell death of the bacterial cell death. And so that is the action by which a lysozyme will be acting as an antibacterial. So this is to see the various mechanisms by which enzymes act. A lot of times, water molecule, is playing a major part in the activity of enzymes. You see it as that donor of protons or hydrogen ions in the reaction. Also, hydroxyl groups from the water molecule, in the formation of products, and also for replenishing, hydrogen ions that are donated from the active center of the enzyme. So that should bring us to the end of looking at examples of the mechanism of action of enzymes, which is in the form of acid-base catalysis. Right. So now we look briefly at enzyme kinetics, and then we look at some applications of enzyme kinetics in enzyme inhibition. Okay, so what is enzyme kinetics about? Previously, I think maybe the first or even the second lecture that we had on enzymes, we looked at some parameters that affect or that influence enzymatic reactions. And some of the parameters we looked at were concentration of the enzyme. We looked at concentration of the substrate. We looked at temperature. We looked at pH. So, I mean, a lot of times these are the parameters that that influence the activity of enzymes or influence enzymatic reactions. So when you study the rate of reaction based on changes in these parameters, what you're doing is you are studying the kinetics of the enzyme. So you in other words, the study of the relationship of the rate of an enzymatic reaction on changes in experimental parameters. That constitutes enzyme kinetics. So if you run enzymatic reactions such that you vary these parameters. So let's take that you are running an enzymatic reaction, and that the product of that enzymatic reaction is a colored product, which means that if you want to study the influence of the concentration of the substrate in that reaction, you have a constant amount of enzyme which is present in the solution, and you have different concentrations of substrates in the solution. So if the product of the reaction is colored, it means that you can actually measure the amount of product which is produced out of the reaction by using a simple method like spectrophotometer, either to measure transmittance or absorbance. So the higher the product which is formed, which means the color is going to be deeper, so absorbance will be higher. Or if it is transmittance, the lower the amount of product which is produced, the higher the transmittance, more light can pass through the solution. So you can do these measurements and have a graph plot, rate of reaction against concentration, or because you measured absorbance or transmittance on your vertical axis, you have absorbance against concentration of the substrate, and you see the relationship, whether it is directly proportional, increasing, or it is reducing. You can do same for pH. So if you run the reaction at different pH values, what will be the effect? If you run it at different temperatures, what will be the effect? You can see the relationship. When you do such, studies, basically what you are doing is you are studying the kinetics of the enzyme. So that is what enzyme kinetics is about. Right. Now there are some models that are used to study enzyme kinetics. And a common model is what is referred to as the Michaelis-Menten equation, which is derived from the saturation curve. This curve we saw earlier when we were looking at the relationship of concentration of substrate with rate of reaction. So rate of reaction on the vertical axis, concentration of the substrate on the horizontal axis. Now, if you remember your senior secondary school core maths, you did a equation of a straight line, which is y is equal to ax plus b, where the a represents the slope or the gradient of the straight line, and then the b is the intersection of the line with the vertical axis. In this case, the vertical axis, the intersection of the straight line with the vertical axis is at the origin, 0. Now, if you write that reaction, you write that equation using these values, where you have y being the V0, which is the rate of the reaction, and the slope or the gradient, if this is the maximum of the reaction, minus say 0. If you find that gradient, and then the x is the concentration of the substrate. If you write that thing, in fact, I'm not going to be asking you to be, deriving any of these things. But what I would like you to keep in mind, you remember, because that is what you may meet if you are doing any enzymatic studies sometime in the future. What you want to remember is that the half of the rate of the half of the maximum rate of reaction, which can be reached. So if you know the maximum rate of the reaction, the half of that, the concentration of substrate at which you achieve that half is what is referred to as the Michaelis-Menten constant. So you see that being written here in this algebraic expression, that Km, that is the Michaelis-Menten constant, when the Michaelis-Menten constant is equal to a substrate, which means that when concentration of the substrate is the Michaelis-Menten constant, at that point, the rate of reaction is half of the maximum rate of the reaction. So you may not be able to find the maximum rate of reaction, sort of experimentally. Say, when you are doing the experiments with enzymes, yes, the graph plateaus this way, but there are slight changes in it, but they are negligible. So you can predict the possible maximum rate of reaction that can be reached with this equation. That if you know the concentration of the substrate at which you can reach half of such maximum rate of reaction, then you'll be able to predict that. Now, this equation has been modified and used in the study of inhibition of enzymes. So, for instance, now we have the Lineweaver-Burk equation, which is a derivative of the Michaelis-Menten equation. You realize the use of this equation will be easier because it's a straight line instead of, this is a saturation curve, and then there has been this estimation from it with a tangent on it. So that's a tangent to have a straight line to do such an estimation. But here you have directly a straight line. And the Lineweaver-Burk equation is just a reciprocal of the Michaelis-Menten equation. So you can see clearly that here, instead of V naught, you have 1 over V naught. And in the Michaelis-Menten equation, you have the denominator being the numerator here. So the reciprocal has been taken. And in the end, you have the equation written in such a way that you can clearly see that, okay, if the equation of the line form that I gave you earlier, which was y equals ax plus b, if this was y, so remember that now the vertical axis is going to be the reciprocal of the rate of reaction, and so it's going to be 1 over v naught. So if you look critically at this, which is supposed to be ax, that is the slope of the reaction, the slope of the straight line, or the gradient of the straight line, times the horizontal axis value. If you look at that, the horizontal axis value is also now the reciprocal of the concentration of the substrate, which is going to be 1 over concentration of substrate, as you see here on the graph. The value there is 1 over concentration of AS on the horizontal axis. If you do that, then it means that the slope is going to be the Michaelis-Menten constant divided by the maximum rate of reaction. Then you can also say that the intersection of the line with the vertical axis is at 1 over the maximum rate of the reaction. So you can easily use this graph. So if you run a reaction in the lab and you vary the concentration of the substrate, and you find the rate of the reaction, and you draw a graph, the Lineweaver-Burk graph, just like this. The moment you draw the graph, the intersection of it with the vertical axis, that becomes the reciprocal of the maximum rate of the reaction. So you can easily determine the maximum rate of the reaction. And you can also easily determine the Michaelis-Menten constant, that is the concentration at which you achieve the maximum rate of reaction from the intersection of the line with the horizontal axis, since that intersection is negative 1 over the Michaelis-Menten constant. So you can use these things to do predictions. These are the two things that are in your course outline to know about, that these two models are used in the study of enzyme kinetics. But you can, I would say, I've written something. I say read on Eadie-Hofstee equation. It's also a derivative of Michaelis-Menten equation. So when you go read on it, even though it is out of your outline, at least broaden your mind, get to know that there is something else apart from these two, which is the Eadie-Hofstee equation. So go read on that and have some extra knowledge on it. All right, so now we'll look at enzyme inhibition. Enzyme inhibition. Of course, we have seen something on enzyme inhibition. We talked of inhibitors of enzymes as substances that either slow down the activity of enzymes, and thus will slow down enzymatic reactions, or they can completely block the activity of an enzyme, which means they can stop or prevent an enzymatic reaction. Now, why is this important? Many biochemical reactions involve the participation of enzymes, and so a lot of pharmaceutical agents are actually based on the knowledge of the activity of enzymes and their inhibition. So that if you know an enzyme which is involved in a specific chemical reaction, and you are able to discover a compound, a chemical substance that can inhibit the activity of the enzyme which is involved in catalyzing that reaction, you can apply that to achieve a certain therapeutic effect. So for instance, you have paracetamol and aspirin that inhibit enzymes that catalyze the synthesis of prostaglandins. And prostaglandins are derivatives of fatty acids, they are hormone-like substances which are participate in processes of the sense of pain. So that if you have paracetamol and aspirin that inhibit enzymes that that catalyze the synthesis of these prostaglandins, then of course it's going to affect the sensation of pain. So that is how these painkillers work. So they actually prevent the synthesis of these hormone-like substances, which are involved in processes that makes us to recognize pain, to feel pain. So we will go ahead and look at some applications of these enzyme kinetics, which we have just studied in enzyme inhibition. Now we start with looking at different forms of enzyme inhibition, and the first is competitive inhibition. So competitive inhibition, from the name, only means that a substance which is an inhibitor competes with a substrate. Then what is it competing for? It is competing for the active center of the enzyme. So we see it being depicted here. On the right-hand side of the screen, you have this diagram where you have an enzyme, which is a round molecule with some portion depicted as the active center of that enzyme. Now the S is the substrate. When S binds to the enzyme, it forms the enzyme substrate complex, and that enzyme substrate complex can then go ahead into a transition state and further to form a product. But you see that there is a molecule I, which looks like the substrate S, but it is not the substrate. You can say it is a structural analog of the substrate. It has a similar structure with it, but it doesn't have the same function as the substrate. And this inhibitor, when it binds to the active center of the enzyme, it forms, call it an enzyme inhibitor complex. And because it's an inhibitor and it is not the substrate that can proceed to the transition state and then form a product, once you have the inhibitor binding to the active site of the enzyme is then blocked from interacting with the substrate. So you see that it is the same active site that the substrate binds to that the inhibitor is bound to. So that is why it's being referred to as a competitive inhibitor. It means the inhibitor comes to compete with the substrate for the active center of the enzyme. You see that what is here in the diagram being depicted in a form of simple equations, sort of a representation of chemical equations where you have E being the enzyme and S being the substrate. So if you have E interacting with the substrate binding to give you ES, which is the enzyme substrate complex, that further goes to the transition state and forms the product P, which dissociates away from the enzyme. But if you look at the reaction which is written vertically, you have the enzyme binding to an inhibitor I, and then it gives you enzyme substrate, enzyme inhibitor complex, which is EI, and that causes a blockade of the enzyme. And so the enzyme is prevented from interacting with the substrate. Now, you look at how enzyme, what we just studied about enzyme kinetics is being depicted here with competitive inhibition. So looking at, so this will be the Lineweaver-Burk, plot of the activity of a competitive inhibitor. Now, what you notice here is that you have I, which is the inhibitor, and the arrow is pointing upwards, which is showing that there is a increase in concentration of the inhibitor. And when you have an increase in the concentration of the inhibitor, you see that the line, the straight lines are also, shifting upwards. But what you notice is that you have an intersection on the vertical axis, which is the maximum rate of reaction, which is not changing. It's the same at all times. So what it means is that for a competitive inhibitor, the maximum rate of reaction is not affected. It is still constant. Why so? Well, it's because, so logically, if, you have an enzyme that an inhibitor binds to and blocks it. If you increase the concentration of the substrate in the enzyme, in that enzymatic reaction, mixture, it is going to increase the probability of the substrate binding to the enzyme. So let me say it again. So you have enzyme and you have inhibitor in a solution. Then you add substrate in that solution. When you continue to increase the concentration of the substrate, the probability of the substrate binding to the enzyme will become higher than the probability of the inhibitor binding to the enzyme. And in fact, even if the inhibitor is already bound to the enzyme, when you continue to add more substrate into the solution, the high concentration of the substrate will cause the displacement or dissociation of the inhibitor from the the enzyme, so that now the substrate takes the place of the inhibitor. And so even though the reaction, the rate of the reaction, the time that you reach the maximum of it will be increased, you will by all means get to that maximum rate of reaction. You can get to it, because it is only one active center that the inhibitor and the substrate are competing for. So you can still reach the maximum rate of the reaction, except that it will take you a longer time because of the activity of the enzyme. And that is what is depicted here. You remember the intersection on the horizontal axis will give you the Michaelis-Menten constant, which is half of the concentration of the substrate which is needed. And you see that as the concentration of the inhibitor is increasing, you also see that the Michaelis-Menten constant is also increasing. So, for instance, this higher line that you see there is now crossing, coming, shifting to the right-hand side. The lowest one is at the farther end. And remember, this is on the negative side of that axis. So as it moves to the right-hand side, it shows increment in the concentration. All right, so you also see what is referred to as uncompetitive inhibition. So in this case, again, from the word uncompetitive, it means there's no competition. So you can see clearly in the diagram being depicted that the substrate S binds to a specific site on the enzyme. That is the active site of the enzyme. But there is a second part of the enzyme that an inhibitor can bind to, which you can refer to as an allosteric site of the enzyme. So the inhibitor is not competing with the substrate for one active site. It has another site that it can bind to. So whether you have substrate binding to the enzyme or not, really, the inhibitor still has a specific site that it can also bind to. No competition. That's why it's being referred to as uncompetitive inhibition. You see it being depicted also in the form of a chemical equation where you have the enzyme E with a substrate binding to give you an enzyme substrate complex. In the normal reaction path, you have the enzyme substrate complex going into a transition state and then producing a product P, which dissociates away from the enzyme E. Now, if you have an inhibitor present in the reaction mixture, that inhibitor I binds to the enzyme substrate complex to give you an enzyme substrate inhibitor complex, ESI, which prevents the reaction from proceeding. And that's what you see being depicted in the diagram. Down here, you have the enzyme binding to the substrate and the inhibitor, giving you the enzyme substrate inhibitor complex, which will prevent the reaction from proceeding further. Either it slows down the reaction or it completely stops it from proceeding. Okay, so here too, we see the Lineweaver-Burk representation of the uncompetitive inhibition, where you see that again, when the concentration of the inhibitor is increasing, this time around you see that the Michaelis-Menten constant also keeps reducing. It's moving to the left-hand side. I mean, what it means is no matter the amount of substrate you are adding in there, it will not change anything, because the inhibitor, it will still have a site to bind to the enzyme. And so the more inhibition, the more inhibitor that you add in there, the more the rate of the reaction, is reduced, and even it is the less amount of substrate that is able to bind to the enzyme to give you any product in the first place. Now, when you also move on, there's also what we refer to as mixed inhibition. Now, mixed inhibition, have two things that can happen, but these two things will result into the same thing, which is the formation of the enzyme substrate inhibitor complex, as we saw earlier, except that two different paths, are occurring at the same time. You see, the normal path of the enzyme enzymatic reaction is that the enzyme with a substrate will give you an enzyme substrate complex, which then gives you the product P dissociated away from the enzyme. Is depicted in the diagram as well, where the enzyme binds to the substrate to give you the enzyme substrate complex. Now, it is either that the enzyme substrate complex is formed before an inhibitor comes to bind to the allosteric site to give you the enzyme substrate inhibitor complex, or first the inhibitor binds to the enzyme, to the allosteric site, but the active center is still available for the substrate to bind to it. So the inhibitor binds first before the substrate comes to bind to give you the enzyme substrate inhibitor complex. Both paths, result into the same thing. Once you have enzyme substrate inhibitor complex formed, the reaction is either prevented from occurring or it is inhibited such that the rate of it is reduced significantly. All right. So you can have these two paths occurring in mixed inhibition. You have it also being, depicted with a Lineweaver-Burk. Here you see that the Michaelis-Menten constant is constant even when you keep increasing the concentration of the inhibitor, but it has shifted to the left-hand side, which means less amount of substrates will be needed to reach the maximum rate of the reaction. And that is also because the maximum rate of the reaction is reduced. Just as you see with the previous one, the think uncompetitive inhibition, you see that there is a shift over there. All right. I mean, don't be so much worried about so far these mathematical representations of these things. I know you can find it difficult to understand it the first time. As a matter of fact, when you are doing these things experimentally, you will understand it, you can get it. But for now, I mean, theoretically, the application of these equations, but it is actually when you run enzymatic reactions, you have results and you plot them. So when you plot them, then you will be able to just determine from the plots the maximum rate of reaction and also see the Michaelis-Menten constant. You just get it from the graph when you have these results and plot them. So it's just like you have some results from your laboratory experiment, and then you plot the graph and you get some intersections on the graph, and you can now determine the value of that intersection, and that value of the intersection is let's say this one, where the intersection of all the lines is the reciprocal of the negative value of the Michaelis-Menten constant. So clearly you can determine the Michaelis-Menten constant from it, which is the concentration of the substrate at which half of the maximum rate of reaction is reached. You can easily do it. All right. Now, so far, what we have been talking about have been, reversible reactions, because, I keep talking of the fact that if you have an inhibitor, but you increase the rate, the concentration of substrate, the substrate will go and displace the inhibitor. That tells you that the inhibition which is occurring is reversible. So even though the inhibitor is having a certain effect, that effect, it's possible to reverse it. So these are reversible inhibitions. Now, the table you see here is just a summary of the things we have looked at so far. So that if you have, so we are looking at the table just brings together the various forms of inhibition which we have looked at, which is competitive, uncompetitive, and mixed inhibition, compared to the reaction without an inhibitor. So the first one is none, meaning no inhibitor in that reaction. You have a maximum rate of reaction, which is Vmax, and you have the Michaelis-Menten constant, which is the concentration of the substrate that's at which you reach half of the maximum of the maximum of the rate of the reaction. Then the first form of inhibition we saw was competitive. And we said the maximum rate of the reaction is not affected because both the substrate and the inhibitor are competing for the same active site. And so if you increase the concentration of the substrate, you can still reach the maximum rate of the reaction, except that the amount of substrate that you need, that is the concentration of the substrate that you need, will be affected. You need more substrate in order to achieve the maximum rate of reaction. And so that change is being depicted here as alpha Km, meaning you need more of the substrate to reach the maximum. The others, you see, yes, they are affected, uncompetitive, and then the mixed inhibition, as we saw earlier, both the maximum rate of reaction and the Michaelis-Menten constant, they are all affected in that case. Right. Now we can look at some possible irreversible inhibition. So irreversible inhibition will mean that once there is that effect of an inhibitor on the enzyme, it is permanent. You cannot, reverse the effect of the inhibitor on the enzyme. So you see being depicted here, irreversible, you have a protein target. That's the enzyme, which is orange, that binds to that protein target, but it can also get dissociated from it. So the arrow is shown in two directions, meaning that it is reversible. Now you can also have the covalent inhibitors, which meaning that the bond which is formed between the enzyme and the substrate is so strong, a covalent bond, that you cannot undo it. And so the effect of that inhibitor on the enzyme is permanent, and actually may even result in the destruction of some functional groups in the enzyme and completely rendering the enzyme inactive in that case. Now, an example of that is what you refer to as DIFP, which is diisopropyl fluorophosphate, and that is an inhibitor of you can use it. This is used in labs to study inhibition of enzymes. This here we are seeing that it is an inhibitor of chymotrypsin, which we saw just a few moments ago. And you saw that in the active center. So what you see on the left-hand side ENZ. So that's the enzyme. And in its active center, we have the serine at the 195th position. We saw this earlier. And you see that there's a hydroxyl group on the side chain of serine. And that hydroxyl group is having interaction with the fluoride of the molecule DIFP. In the end, you see that the enzyme is forming a covalent bond with the molecule. And that covalent bond, it's sort of, this is an ester bond, phosphoester bond. The phosphates in the DIFP is bonded covalently to that serine side chain of the enzyme. That is making it, rendering it, completely inactive. Now, I think if you look at the labels of some insecticides, for instance, you will see DIFP, or some molecules, some compounds that are fluorophosphates, and these fluorophosphates, can bind to, for instance, a neurotransmitter, say. A common one which we saw earlier was acetyl, choline esterase, choline esterase. And you remember that we talk of the acetylcholine esterase to be involved in muscle contraction and relaxation. Now, how this DIFP will be acting in insecticides is that, since it can bind to, say, the enzyme acetylcholine esterase, it means that it will prevent that activity of the muscle contraction and relaxation, the transfer of signals to the muscles to move. And so in the end, what it means is that you have the DIFP acting in a way that it causes paralysis in the organism. So once it blocks the activity of acetylcholine esterase, and in fact, if it is in such high quantities, in high concentration, it goes in there and binds permanently to the enzyme and prevents the enzyme from acting in the process of transmitting signals that results into movement of the parts of the organism, rendering the organism paralyzed. So that is how you use. That's an application of the knowledge of inhibition, enzyme inhibition in the paralysis of pests, for instance. All right. I think this is also on irreversible inhibition of the enzyme glycopeptide transpeptidase. And glycopeptide transpeptidase is involved in the formation of what we saw earlier, the peptidoglycan cell walls of bacteria. So the enzyme glycopeptide transpeptidase, the name even tells you that it is involved in the transfer of the monomers that will come together to bind to give you that cell wall. It's a transpeptidase, and at the active center of this enzyme, the glycopeptide transpeptidase, also there you have serine, and so it's depicted the active center having the hydroxyl group. Now, penicillin, acting as an antibiotic, can act on this glycopeptide transpeptidase such that the penicillin molecule, which is depicted here, is now bound on that the serine residue, which is at the active center of the enzyme glycopeptide transpeptidase. And once it binds to the enzyme permanently this way, it means that the enzyme is rendered inactive. If the enzyme is rendered inactive, it means there cannot be the formation of the peptidoglycan, which forms the cell walls of the bacteria. In other words, the cell walls of the bacteria is going to get destroyed, and that results into the cell death of the bacteria. So that is how penicillin can also act. Some of these irreversible inhibitors are referred to as suicide inactivators. I mean, they can cause some sort of apoptotic reactions in cells. I think I refer to this, if not with you, maybe with MLT students, where I mentioned the process of apoptosis that can occur in cells, where you have, let's say, for instance, cancer cells recognizing that they pose danger to other cells, and so they begin to initiate their own death so that they don't infect others. So there is that sort of protective mechanism within cells, to protect other cells, where they actually initiate reactions that cause their own death, being referred to as apoptosis. Right. I think so. This is the end of today's session. I think now I'll open the floor if you have any contributions, any questions, any comments you can make before we close. So the floor is open. If you have anything, you can put up your hand, and I'll call you. You unmute yourself and you speak.